Thank you, sir. I feel very good and great to hearing about myself from my guide's mouth. So, uh, good morning, one and all. Good morning to my respected guide, Dr. N.M. Dongarwar. All my respected teachers who are there as a participant for this refresher course, my colleagues, and remaining faculty friends. So, I'm Dr. Uma Thakur, working as assistant professor in Institute of Science, Nagpur. And I will try to talk something about ex-situ conservation of some orchids. You might have heard about this topic from expertise mouth, always from Dr. Dongarwar's mouth. But today I'll dare to speak on this topic. So let us start with the lecture. So my topic is ex-situ conservation of some orchids. So as per Orchidaceae is concerned, orchid flowers carries a royalty in its features or appearance. Among the flowering plants, they comes under most intelligent, beautiful, and patient class. Why I'm calling them as intelligent? Because they have adapted themselves. They are super specialized in certain factors. So because of this, I believe that they are the most intelligent flowers on the earth and beautiful because the flowers are very beautiful, and the flowers are patient one also because they bloom once the scape or inflorescence of the orchidaceae flowers blooms. They can wait for 1 month or more than 1 month to wait their specific pollinator to pollinate the flower. So as per Orchidaceae is concerned, it is the largest and most diverse group among the angiosperm. Lots and lots of diversity is there among the angiosperm. And as per Royal Botanical Garden Q, 880 genera is there and which are distributed in 22,000 species. In some of the paper, I have also seen that 25,000 species are there. As per India's concerned, India is blessed with the wealth of 1,300 species, and in Maharashtra, Orchidaceae is represented by 110 species, which are distributed in 34 genera. We all know that the biodiversity is at a major threat nowadays. Many and many plants are entering into red data book, and the major reasons for them are that they maybe all those plants which are entering into red data book, they are overexploited. But as for Orchidaceae is concerned, this is the most advanced family among the angiosperm. So that's why we place this Orchidaceae family in monocots, and that to be at the end of monocots because of its advancement. No doubt, the family is more adapted to its habitat, the family is more advanced, but what is the problem with the family? Why it is entering into IUCN's red list? So let us discuss about this, that what are the major factors which are responsible for a regular decline in the family. So when we have started doing ex situ conservation of Orchidaceae, the first question that came to our mind is, what is the need of conservation of this family? No doubt the family is well advanced. It is well adapted to its habitat. But when something goes over, then it is a problem. In this situation, the plants grow in a specific habitat. They need a specific habitat for its survival. Then another thing is that the flower is very complex. The flower is very complex that not even a honeybee or sometime an usual wasp will come and pollinate the flower. They need a specific pollinator for its pollination. Specific habitat is required, specific pollination, specific pollinator is required for the pollination. Another thing is that the seeds are millions of seeds are encapsulated in a single capsule. They are very much like a dust particle. They are microscopic. And another thing is that they are non-endospermic. Non-endospermic seeds means they always need a symbiotic partner to provide nutrition in the early age of its germination. So again, it is a problem. And no doubt, there are millions of seeds produced by a single capsule, and on a scale, 10 to 12 capsules are there. So you can calculate that 14, 15 millions of seeds are produced by a single plant, orchid plant, in one season. Then another problem with this family is symbiotic seed germination. The seeds are non-endospermic. They can't germinate at its own. The initial nutrition is always given by the symbiotic partner. So symbiotic seed germination is there. Another problem with this family is that the plants are very, very slow growers. Follow C4 as well as CAM pathways. So they are very, very slow growers that even though we'll discuss it in the second half of the lecture, that the plants take 1 hour to complete into a complete seedling. Okay, so they are very much slow grower. So because of these factors, like specific habitats, specific pollination, seeds are microscopic, they are dependent on other. Because of these reasons, the plants are entering into red data book. Not only these are the factors, these are certain internal factors. Suppose if we start dividing the factors which are responsible for declining in the biodiversity of this Orchidaceae family is that, so we can categorize them as external factors which are coming from the human beings, we can say directly, and some factors which are there in the family itself, like intrinsic factors. So when we are considering intrinsic factor, it means the family is most advanced one. They need a specific habitat for the growth. The flower is complex, that not any pollinator will come and pollinate the flower. They need a specific pollinator for the pollination. Seeds are dependent on others for their initial nutritional need. And next one is they are slow growers. And if we talk about the external factors, all those factors which are anthropogenic activities, they are also playing important role to entering these plants into endangered list. So let us discuss each and every point one by one. The first important point about the Orchidaceae is that they need a very specific habitat for their germination, for their growth. We usually divide the habitat in two major categories. One is terrestrial habitat and second one is epiphytic habitats. It means some of the orchid species, they grow on the land, so that's why they are called as terrestrial orchids. And some of the plants, they have evolved, they have climbed up the trees, so they are in epiphytic habitats. So if the plant is epiphyte, it is not a parasitic one. The plant epiphytic means that they are dependent for the bark, or they usually grow on all those plants which have thick bark for their moisture need. So the plants are not parasitic, they are epiphytes. So basically two types of habitats are there. One is epiphytic habitat. So that's why all those plants which are growing on the trees called as epiphytic orchids. And second one is the terrestrial orchids. So when we talk about the terrestrial habitats, again, there is certain range of habitats where usually we found the orchids. Even though some of the plants, they grow in the lithophytic habitat, it means that the rocks are somewhat covered by a layer of soil, and that habitat is suitable for some of the orchids. And some of the plants, they grow in a wetland position, that here in this picture you can see there is a wetland. We have collected one plant, that is Zeuxin stratomitica, which is only and only present in this habitat. So we can call that Zeuxin stratomitica is a wetland plant. So even though plants are growing on the trees like epiphytic one, some are growing as a terrestrial, but still in these two categories, they are again very specialized about their habitat. Let us discuss some of the orchids one by one. So this is a plant called as Geodorum densiflorum. This is a terrestrial orchid, and we have collected this orchid only in a specific range of forest area. These plants are usually grown there. They are very slow growers. If weeds will come into that territory, so these plants may not tolerate that situation. So here in this picture, you can see that these plants are growing very nicely under the cover of trees. So they are mostly growing in the lower strata of the forest where weeds are not there. Then this is the another plant, Pectalis gigantea. This is the largest flower among the orchidaceae in India. And this plant is mostly present in the Kolhapur or in the western Ghats of India. Then again, it is habitat specific. The plant is habitat specific. This plant, we have not collected not even a single individual of this plant in Vidarbha region. Then this is another plant called as Narvelia araguana. This is the leaf is heart shaped, and again this plant is growing under the cover of forest. So there is very rich humus. You can see the condition of the soil. The soil is very rich in humus, and this plant is again restricted to certain area.
Excuse me, ma'am. I think your
Yes,
voice
sir.
is breaking, ma'am.
Um,
Sometimes your voice is breaking.
so shall I remove my headphones if you feel so? Can I try that?
Uh,
Yes,
okay,
ma'am.
ma'am. Okay.
Yes.
Okay.
Let me try this.
Ma'am, is there any problem of network at your end? अभी आपकी आवाज नहीं आ रही मैम. आपकी आवाज बिल्कुल सुनाई नहीं दे रही अभी. Ma'am, you can use your headphones.
Sir, am I audible?
Yes, ma'am.
Yes. Now it is audible, madam.
Yes, it is clear now.
Yes, your voice is coming. Your
Sir,
voice is
maybe
audible
because
now.
of certain issues that voice is disturbing in
Okay, no
between.
problem. No problem, ma'am.
Okay.
You
So
can
shall I
continue.
start? Okay. Thank you, sir.
Yes, ma'am.
Okay. So this is the plant Narvelia araguana. The heart-shaped leaf is there. And if you observe the soil structure, you can see that this plant is growing in very rich humus soil. Then okay, so this is another terrestrial habitat. Till now, we have discussed that the plants are growing under the area of forest cover. Now, this is the open grassland area. Again, it is a terrestrial habitat of Habaneria grandifloriformis. You can see the white dots are here. These are the scapes of Habaneria grandifloriformis. And this is the open grassland where this plant is growing luxuriantly. Then this is the plant of Habaneria grandifloriformis, a very small single leaf and a single flower is there in this plant. Then this is another terrestrial plant orchid that is Habaneria longicorniculata. We have observed this plant only in the ranges of Sahyadri Hills and in the grasslands of Sahyadri Hills. If you observe this plant, there is a very long spur in this plant. So only those pollinators will come and visit this flower, which will have this much long proboscis, and mostly the moths are have this much long proboscis, and only specific pollinator will come and visit this plant. Here you can see there is a very long proboscis, and long spur is there, and only those pollinators will visit which have this much long proboscis. Okay, so again this plant is growing in open grassland area, but where the grasses are at its very high, long grasses are there. In the previous picture, we have seen that that is the terrestrial habitat, but the height of the grass is almost of 1 feet. We can say almost 1 feet, but here the grass height is almost 4 to 5 feet. Then this is Habaneria marginata. This is, we can say that the plant which are growing in the Vidarbha region. This is, this plant we have collected Habaneria marginata at various places in the Vidarbha region, but very limited number is observed in the Western Ghats. So this is Habaneria hainiana. You can see again, this is specific habitat of Habaneria hainiana. You can see different scapes are there. And this is again a specific habitat for Habaneria hainiana, and we have found the plant Habaneria hainiana in Western Ghat ranges. Then this is Euxine stratimatica growing in the wetland area. Again, this plant is collected from one of the wetland of Umre district, and we have collected this plant only at one site. We have observed the specific localities also for this plant, but we have collected the plant only from one specific locality. Then let us move to the epiphytic habitat. Here you can see that this is the tree which is having a thick bark, and Venda tessellata is growing very luxuriantly. You can see the bunches of Venda tessellata is there. Then this is the flower of Venda tessellata. The same habitat is shared by Venda testacea. No doubt, Venda tessellata and testacea are belonging to same genera, but still the problem is that we have collected Venda tessellata from the adjoining vicinity of Nagpur area, but this plant Venda testacea is only and only growing in the Medghat region, and only in a specific locality, that is only a small area is there where this plant is growing. Then this is Arid maculosum, again epiphytic orchid. Then this is the plant of Arid maculosum. This is Rincostylis retusa, again epiphytic orchid. Venda testacea, Venda tessellata, then Arid maculosum, then Rincostylis retusa. The needs of all these plants are somewhat similar, and they usually grow in such localities. This is a locality near to Balaghat region where mango evergreen plantation is around the area. And if you see that adjoining vicinity of this mango trees, there is a paddy field. So because of the paddy fields, there is lots and lots of moisture is there in this area. So these plants are fully rich in various species of orchids. So this is another epiphytic orchid specific to Kolhapur and Western Ghat region. This is Oberonia recurva. You can see that this plant is growing in a very high moisture condition along with the mosses. Then this is a species of Dendrobium. Again, we have collected it from the Kolhapur region. This is Iria dalzalai. So you can see that this is the correct habitat where these epiphytes are growing, very rich in moisture. This is the picture of Western Ghats. And this is a lithophytic habitat of Habaneria rariflora. This is a very rare habitat. Here you can see on the rock a small layer of soil is there, and on that habitat this plant is growing. So till now we have discussed that the plant needs a specific habitat. Then we have discussed that in those specific habitat, still there is again specificities that some plants are growing under the forest cover, some plants are growing in open grassland, some plants are growing along with the long grasses, and then we have discussed about the specific habitats of epiphytes. So no doubt they need a specific habitat. This is an internal factor for the decline of the species. But along with the internal factor, how we can forget the external factors? So here comes the role of humans. So major threats given by humans is the habitat destruction.
Excuse me, ma'am.
Yes,
मैम आपकी वॉइस ना ब्रेक हो रही है, स्टार्टिंग से ही हो रही है, तो आप थोड़ा वीडियो ऑफ करके देखिए कि वॉइस क्लैरिटी आती है क्या.
ma'am, let me change my position. Give me one second.
पार्टिसिपेंट्स आपको वॉइस क्लियर आ रही है क्या कि मेरे तरफ कुछ इशू है?
नहीं मैम, क्लियर नहीं आ रही.
No,
No
madam,
madam,
same here, same here.
Yes.
ओके 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 या
Take a second, ma'am. Give me one second. Let me change the position near to Wi-Fi and you open the windows.
Just off your video.
हाँ यस मैम, वीडियो ऑफ करके एक बार देखिए, मे बी वॉइस।
Even the PC is going very slow because of
उमा मैडम,
yes sir.
सिर्फ वीडियो ऑफ कर दीजिए, बाकी हो जाएगा शायद।
सर मैं ट्राई कर रही हूँ बट सिस्टम में कुछ भी प्रॉब्लम चल रहा है।
ओके ओके ओके।
सिंबल
नहीं मैम, सर उनके सिस्टम में कुछ इशू है।
The system is taking too much long time to respond.
Yes ma'am, no issues, please continue. If possible, keep your video off so that voice clarity will be there. You can
Okay,
continue
let me
ma'am,
try that.
continue.
Now the voice is clear?
मैम थोड़ा सा तो है ही डिस्टरबेंस, बट देखिए कंटिन्यू करिए, नो
Ma'am,
इश्यूज।
even I feel the same disturbance from your end, that
अच्छा,
distractive
maybe
voices.
it may be the
Sometimes it comes in between. Sometimes
Yeah.
the voice is clear and sometimes.
okay okay. Yes ma'am, please continue.
Okay. So here comes the role of humans, and this is the photograph of Meghat region, and from this picture it is very much clear that zoom cultivation is there. The forest is cleared by the farmers for the growth of crop. Basically, this is the habitat of Habaneria grandifloriformis. We have collected the Habaneria grandifloriformis from this region. And when we visited the region in the next season, again was we have visited the same area in the next season. We have found that all the plant or population of Habaneria grandifloriformis was wiped out because of this cultivation. Now the slides are not moving.
मैडम शायद डिस्कनेक्ट हो गए हैं, प्लीज वेट।
ओके मैडम।
मैडम ज्वाइन करते हैं, तब तक मुझे कुछ इंस्ट्रक्शंस देनी है। 39 लोगों के ही पीपीटीज आए हैं। बाकी पार्टिसिपेंट्स अगर सबमिट नहीं किया होगा तो प्लीज सबमिट करिए सेमिनार के पीपीटीज। Only 39 participants have submitted their PowerPoint presentations.
हेलो, गुड मॉर्निंग प्रीति मैम।
Yes, good morning, sir.
मैम, आपने जो एक जो वीडियो सेशन आपने रखा था क्वेश्चनर वाला, फीडबैक वाला,
हाँ
तो वो हमने असाइनमेंट प्रिपेयर किया था, पर उसका लिंक शायद अपलोड के लिए नहीं दिखा सर।
सर था ना कल के सेशन में, कल के डेट में।
उसमें मैडम जो है ना एक क्विज का था।
हाँ हाँ सर, उसके ऊपर नहीं दिया था, गूगल फॉर्म के ऊपर मैंने असाइनमेंट नहीं दिया था, सिर्फ वो गूगल फॉर्म प्रिपेयर कैसे करते हैं वो दिया था। और क्विज मेकिंग के ऊपर असाइनमेंट था।
हाँ हाँ हाँ, क्योंकि वो
हाँ
गूगल फॉर्म वाला वीडियो जब खत्म होने को आया तो आपने बोला था कि
हाँ
आपको
यस यस, लेकिन उस दिन बहुत असाइनमेंट्स हो चुके थे ना, इसलिए मैंने वो नहीं दिया था। ऑलरेडी 2 असाइनमेंट, एक रिपोर्ट और एक क्विज था, प्लस आपका सेमिनार का भी प्रिपरेशन था, तो इसलिए मैंने उसके ऊपर नहीं दिया था असाइनमेंट।
अच्छा जी मैम,
जी
थैंक यू।
ओके।
प्रीती मॅम,
हाँ
हा मॅम, आम्ही सेमिनारसाठी जे पीपीटी सेंड केलेले आहेत त्याच्यामध्ये थोडंसं एडिट करून आम्ही प्रेझेंट केलं तर चालेल ना?
हो, तुम्ही तुमच्याकडनं शेअर करणार आहेत का स्क्रीन?
हो.
ओके ठीक आहे, कारण की बाकी सगळ्या पार्टिसिपेंट्सला मी सांगते की आय विल शेयर द स्क्रीन सो दैट यू कैन एक्सप्लेन फ्रॉम यूर एंड फ्रॉम मोबाइल ऑल्सो। किसी को अगर अपने तरफ से शेयर करनी होगी लैपटॉप की स्क्रीन तो वैसे आप कर सकते हैं।
प्रीती मॅडम,
अच्छा, नाहीतर मग
Yes.
तेच प्रेझेंट करावं लागेल जे आम्ही
नाही,
सबमिट
तुम्ही
केलेलं
हॅलो.
आहे.
परत पाठवून द्याल ना मॅडम, करेक्टेड वाला जे असेल ते परत पाठवून द्याल।
ओके मॅम, ओके.
मॅडम
प्रीती
मी
मॅडम,
वसुधा बोलते
हा
मॅम. माझ्या सेमिनारच्या वेळेस तुम्हीच स्क्रीन शेअर कराल मॅडम, कारण मी मोबाईलवरच करणार आहे.
हाँ मेजोरिटी ऑफ द पार्टिसिपेंट्स के लिए मैं ही शेयर करूंगी, बट किसी को कुछ स्पेसिफिकली और कुछ करना होगा, कुछ एनिमेशंस वगैरह होंगे, तो अगर इच्छा होगी तो आप अपने तरफ से भी कर सकते हैं, बट फॉर ऑल द पार्टिसिपेंट्स आई विल शेयर सो दैट वी कैन सेव अवर टाइम।
ओके मॅडम, इट विल बी मोर बेटर मॅडम.
यस यस, नो इश्यूज।
मॅडम
हॅलो.
हाँ
प्रीती मॅडम,
हाँ बोला।
उमा मॅडम 5 मिनिटात जॉईन करताय, डोंगरवर
हो
सरांचा
हो
लॅपटॉप बोलणं.
हो, दिसला मला मेसेज दिसला।
ठीक आहे.
Hello, madam.
ओके 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 सर, ओके।
Hello, ma'am.
हाँ बोलिए मैम।
Madam, I have submitted PDF of PPT
So we were discussing about the threat which are because of anthropogenic activities. यस कहाँ से यहाँ से ना यस हाँ स्टक हो गया था Am I audible?
Yes, ma'am.
Okay. So this is the
Yes, ma'am.
habitat of Habaneria grandifloriformis near the Medghat region, and we have found multiple population of this plant here. But when we have visited twice and thrice the same habitat, because of the cultivation, which is a cultivation activity, agriculture widening, deforestation of that area, that population of Habaneria grandifloriformis was wiped out completely from that area. Then another thing is that this is the habitat of Zeuxin statomatica. Here you can see heavy grazing is there. Because of grazing, the plants are, the number of the plants are eaten by the animals. This is the habitat of Habaneria panchganensis, critically endangered orchid, eaten by horse. Again, this is the Panchgani Plateau. This is a habitat of Habaneria panchganensis as well as Habaneria rariflora. And grazing is the major threat in these localities. Another thing is that this is the habitat of Habaneria rariflora, where you can see that road widening is there. This was earlier the picture we have seen Habaneria heniana in this area. Because of the widening of this hotel, the complete plot was later on when we have visited the plot. Again, we have seen that the whole area was already allotted to a hotel construction. So again, road widening as well as construction, these are again important factors which are responsible for the declining the number of the plants. This is a Panchgani Plateau because of the tourism. Earlier the name Habaneria panchganensis, which is a critically endangered orchid. The name Panchgani, because of the Panchgani Plateau, the name was given to the plant Habaneria panchganensis. But nowadays, if you visit the same plateau, you will see that not even a single individual of Habaneria panchganensis is there because of tourism. The lower two pictures belonging to Karst Plateau. Now the tourism is permissible into that area also, and because of human intervention, because of human activities, many species are wiping out from that area because of the tourism. Till now, we have discussed about the specific habitat, the threat because of specific habitat. These are certain plants which are growing in our area, or these are the plants which we have observed in our investigation, and we found that this is the status of these species, that Aridis crispum is vulnerable, Aridis maculosum endangered, then Dendrobium barbaculum is vulnerable, then Habaneria, many species of Habaneria, they are critically endangered, near threatened. So this is the status, current status of these species. So let us move to our what we have did in this investigation. In this investigation, we have visited Maharashtra state, Madhya Pradesh, Karnataka, Tamil Nadu, Arunachal Pradesh, and Himachal Pradesh for searching the population of Habaneria. In spite of moving for in situ conservation, because there is a technical problem in this in situ conservation, that you can't protect each and every habitat by declaring it as a biosphere reserve or something national park like that. So here we have shifted to ex situ conservation. We have collected some of the plant and tried to adapt these plant in greenhouses and tried to multiply them, and later on we have tried the eco restoration of these plants. So in this investigation, we have collected 53 orchid species. These are some of them, like this is Acanth, then Arids maculosum, Cotonia, Geodorum densiflorum, then this is Habaneria plantagenia, this is a Habaneria species, Habaneria ovalifolia, Habaneria comilinifolia, Habaneria elizabethae, Habaneria rariflora, Habaneria foetida, Habaneria foliosa, then Habaneria marginata, Habaneria roxburghi, Habaneria hainiana, Habaneria panchganensis, this is a critically endangered orchid, then Dendrobium barbaculum, then Iria dalzelae, Liparis, Melexis, then Oberonia recurva. This is another species of Melexis redi. Then Peristylus guderoides, Peristylus loi, Peristylus densus, Venda tessellata, Luisia gelanica, then Foetida. Nervalia araguana, this is Rhincostylis retusa, Venda testacea, and Zeuxin statumetica. These were the pictures of the plants which we have collected in this investigation. So first we have collected the plants, and then we have tried to provide them all those specificities which were already there in the nature. So we have tried to acclimatize all the epiphytes here in the pots along with the coconut husk to give that moisture as well as the nitrogen content from the coconut husk. These are the Habaneria foliosa plants. You can see how luxuriantly they are growing in our greenhouse in the next season. Then these are the plants. Here you can see the flowering is there in all those plants which were adapted to this habitat. Habaneria rariflora which were growing along with the rocks. So we have tried to maintain that condition also. So we have replanted the plant along with the pebbles and rocks. So the next point in this was, first was specific habitat. Then let us talk about the complexity in the orchid flower. So orchid flower is having 3 sepals. These are 3 sepals and these are 3 petals. One of the petal is modified into labellum, which is actually acting as an attractant for the pollinator. This labellum is a lip-like portion which is attracting humans as well as all the pollinators. Here male and female reproductive systems, they are fused together into a column, and in this, here you can see in the all remaining angiosperm, you usually found the pollen. Here pollens are in the fused form. We call them as pollinia, and this is the stigma portion. So this is the stigma portion of this column. Here, if you take the pollinia and if you insert that pollinia in the stigma portion, this is the actual process, or this is the method how we can achieve pollination. So no doubt, animals are brained. We all believe that we can think, we can manipulate the things, we can think anything which is, we can achieve anything which is in our mind. But plants are not very much, we can say that they are not very much used to this, but still they are very much adaptive to its habitat. They are very much specialized. In the first slide, we have discussed that plants are very much specialized. Here you can see this is a orchid plant, orchid flower. Here, this is the length of the spur, and this is a specific pollinator which is having this much long proboscis. It means that this Disa plant will always be pollinated by only this pollinator which here have the same spur length as the proboscis length. So whenever this moth visits the flower for the sake of nectar, then at the time of sucking the nectar, these pollinias, you can see the pollinias are attached to the proboscis, and when the same pollinator visits the second flower, then unknowingly this pollinator will perform the process of pollination. So you can see how specialized pollination is there in Orchidaceae family. Another well-known example of sexual deceit is there in the in the orchid pollination is there. So this wasp is a pollinator of this ophris flower, and if you see the labellum of this ophris flower, this labellum is showing the similar appearance to the female partner of this male wasp. This is a male wasp thinking that this labellum is a female partner. So whenever these wasps will come out, immature wasp will think at the same time this orchid flower will bloom. Immature male wasp will think that this is the female partner, and in this species of wasp, the female partner is wingless. So they go for air mating. The male partner carries the female partner in the air for air mating. Since the labellum of orchid is similar to the female partner of this wasp, the immature wasp will think that this is the female partner, and unknowingly he tries to pull out the labellum of the orchid for the air mating. In this process, the pollinia is attached to the upper body of this wasp, and whenever the same wasp visits the another ophris flower and thinking that now this is the female partner, unknowingly this wasp will do the pollination. So here we can say that how clever that ophris plant is, that he adapted the same structure of the labellum as that is there, the structure of the female partner of this wasp. So this is called as sexual deceit pollination in the Orchidaceae. So this is a specific pollination, and if you take different examples of Orchidaceae family, in every plant you will see different mechanism is there as per pollination in this family is concerned. Every time you will see a different condition, a different mechanism is adapted by the plant. Then what we have did, no doubt we can't be a pollinator practically, but still we have tried artificial pollination in the absence of pollinator. Since pollinator was absent in our area, so we have started becoming a pollinator and tried artificial pollination. So here first thing is that the first challenging thing is that what is the male part in that specific flower and what is the female part. So here you can see the two eye-like structure. These two eye-like structure on the apex of the column, these are the pollinias. And if you pull out the pollen cap, and below this here you can see, if we pull out the pollen cap, pollinia is there, and this portion just beneath the pollen cap, a dentation or a hole like structure is there, which is actually the stigma, sticky stigma portion. So if you take out the pollinia and if you stick that pollinia here, then this is the process of artificial pollination in the family Orchidaceae that we have tried with the multiple species where the natural pollinator was absent in our area. This is Pathoglottis plicata, one of the orchid. We have tried artificial pollination, and here you can see that artificial pollination was successfully achieved, and we have achieved a success rate by developing the capsules. This is Venda tessellata. Artificial pollination was tried in Venda tessellata, and you can see the capsules were developed out of self as well as cross pollination. Then we have also tried the pollination in the nature, that whenever there is a very limited number of wasp, here you can see that this is the photograph of Habaneria species, especially Habaneria roxburghi. You can see a long spur is there, and this plant is always visited by a moth which will have this much long proboscis. It means that specific pollinator is required for the pollination. So whenever we used to visit to the nature for the collection, we have also tried artificial pollination in the nature. This is a flower of Habaneria marginata. We have tried artificial pollination in our Habaneria marginata also. This is Dendrobium barbaculum. You can see how much healthy capsules were developed out of this, the artificial pollination that we have tried. In this plant here, you can see that adaptation is there. The spur-like basically the hairs are there on The labellum portion is again modified with the small hairs. These are again attractant. It seems like that there are number of pollens at this point of time, so that multiple wasp, multiple bees can visit for the sake of pollens. And but now these are just hairs, they are not pollens. So here the plant is defeating the pollinator for the sake of pollens. So we have tried artificial pollination and we have achieved very good results. Here you can see that capsules are developing. The plants are acclimatized well in our poly houses, so that's why we are getting nice flowering in Dendrobium barbatulum. Here you can see the result 90, 94, 83. These are the results of consecutive years where we have tried self as well as cross pollination in Dendrobium barbatulum. Then we have also tried artificial pollination in Geodorum densiflorum, which is a terrestrial orchid. And here you can see that out of here you can see 4 and 5 flowers are there in one scape. Here you can see the 100%, almost 100% capsule development is there out of artificial pollination. Then we have compared in some of the plant, we have successfully done the artificial pollination, but in some of the plant they are more and more adaptive to their pollinators, so we failed to do artificial pollination. So this is the comparison of pollination percentage in the nature and pollination percentage which we have tried in our poly house. So here you can see, if you see the Zeuxin stratomatica, almost somewhat similar results we have found. But in some of the plants like Nervalia araguana, like Liparis difflexa, and in some of the cases we failed to achieve that success rate. Maybe they are very much specific about their pollinator and we failed to achieve that pollination percentage which are there in the nature. So this is the comparison of artificial pollination as well as pollination which is happening in the nature. So this was the second specificity. We have tried to overcome the second specificity of Orchidaceae family, that we can try to achieve the number of seeds out of these capsules which we have adapted or acclimatized in our polyhouses. Then the third specificity of the family is seed specificity. No doubt the scape is very patient. The orchid scape is very patient. The flower waits for their pollinator for so long, and if the pollinator visits the, you can see the capsule is developed in a single capsule. Millions of seeds are there. And if you see, this is the scape of Habaneria panchagonensis, critically endangered orchid. Here 3 and 4 capsules or 5 capsules are developed in one scape. It means that this much million of seed was produced by a single plant. So if this much number of seeds are produced, then we should not think about the conservation. Then it means that you will see everywhere orchid plant is growing, because millions of seeds are encapsulated in a single capsule. But again, this is the family Orchidaceae, so they are very specific. If you see, this is a microscopic structure of the seed. I have taken this picture under the microscope 10x. Here you can see only this is the embryo portion, and it is encapsulated by a layer of testa. So here this testa is, the first thing is that testa is a very minute thin layer over the embryo, and the seeds are non-endospermic. If the seeds are non-endospermic, we all know that endosperm provides the initial nutrition for the seed for the sake of germination. In this situation, the seeds are non-endospermic. It means that initial nutrition is not stored in the seed. Then this seed is totally dependent upon its symbiotic partner. So here in Orchidaceae, seeds usually germinate when it makes an association with a fungal partner. So what the fungal partner will give? Initially, the fungal partner will release some of the cytokinins as well as some of the nutrition is provided by the fungal partner, and the germination of orchid seed will start. So that's why the germination is called as symbiotic seed germination. So when we have tried seed germination in the laboratory, the major problem was that the testa is highly lignified. So whenever we used to keep these plants, or whenever we used to keep these seeds in the vicinity of media, so these seeds were, because the seeds are lignified, they fail to interact with the water, they fail to interact with the media. So again, there was a problem. So we have overcome the problem by giving some bleaching agent to the seeds, so that the lignin component will be delignified and the seeds can interact with the media for the germination. Here you can see the multiple seeds are there. This is the microscopic picture. So in present investigation, we have divided our tissue culture experiments into media standardization, then asymbiotic seed germination, because here the symbiotic partner is missing. So that's why the germination is called as asymbiotic seed germination, and third one is micropropagation. So for this, we have tried 6 different medias to know what is the nutritional need of that specific seed for the germination. 6 different medias like BM1 media, then Knudsen C media, LM media, MM media, Murashige Skog media, and Wesson-Wendt media. So for each seed, we have tried different concentrations of cytokinins as well as the auxins, along with the 6 different medias. So let us see the results. So these are the results of Peristylus loi. In Peristylus loi, we have tried 6 different medias for the sake of to know the nutritional need in a better way, which media is supporting highest rate in the seed germination. So this is the Peristylus loi. In Peristylus loi, you can see these are the initial germination, then the complete plant was developed, then that plant, once the plant was germinated, then that plant was also tried for the variable concentration of sugars. So variable concentration of sugars were tried with the terrestrial orchids to increase the size of the tuber. So here, when we have tried variable concentration of the sugars, so that the size of the tuber will increase. So why we want to increase the size of the tuber? Because we know that tissue culture plant is not that much capable, or that tissue culture plant is not, or it takes lots and lots of care to stabilize in the nature. So if you convert that plant, or if you store the energy of the plant in the form of tuber, if you can increase the health of the tuber, it means that the next, whenever you incorporate this tuber into the soil, this tuber will grow immediately in the next season in a new plant. So here the success rate of reintroduction is increased if you increase the size of the tuber. So we have also tried, along with the 6 different medias, variable concentration of the cytokinins and auxins. We have also tried variable concentrations of sugars, because we all know that tubers are basically capable to store the sugars. So we have increased the sugar concentration to raise or to increase the size and health of the tuber. So this is the asymbiotic seed germination of Peristylus loi. Then these are the variable photographs of Geodorum densiflorum. Geodorum densiflorum, again a terrestrial orchid. So asymbiotic seed germination was tried along with the 6 different medias, and here you can see what seeds are inoculated on the media. Seeds are inoculated, then seeds have started germination. You can see the complete plant here. So this is the germination in variable medias, that is Geodorum densiflorum. Then this is Cymbidium cymbidium. Here you can see the growth of Cymbidium. Then next one is Oberonia recurva. in the slides, we have seen that Oberonia recurva was growing in the nature along with the mosses. So when we have in vitro multiplied the plant by asymbiotic seed germination, you can see how nicely the seeds are germinating in different medias. So we have tried to incorporate the same thing when we were trying to acclimatize these plants in the nature. So these plants were acclimatized on the mosses, Oberonia recurva. Then this is Acanth Primosa, tiger orchid. You can see the rate of germination is very high when we have tried the different concentrations of cytokinin in the variable concentrations of or different types of medias. Then this is Arids maculosum. You can see the germination or asymbiotic seed germination, how luxuriantly the Arids maculosum is growing. Then this is the Dendrobium barbaculum. You can see variable stages of germination of Dendrobium barbaculum. Then this is Eulopia nuda. Eulopia nuda, we usually call it as a amarkand because of the anti-cancerous properties of Eulopia nuda. The corms are basically used. So if you want to know more about Eulopia nuda, is that Eulopia nuda as well as Geodorum densiflorum, both the plants look somewhat similar in the nature. The morphology is somewhat different. If the plant flowers, then you can easily identify between the Geodorum as well as in the Eulopia nuda. But if you see the plant in its corm stage, then the corms are somewhat similar. So mostly Eulopia nuda is collected by the tribal people because it acts as because of its anti-cancerous properties. We civilized people hired the tribal people for the collection of the corms of Eulopia nuda because of its anti-cancerous properties. So along with this Eulopia nuda, because the corm of Eulopia nuda as well as Geodorum densiflorum both are similar, so the plant of Geodorum densiflorum, which can usually luxuriantly grow on the forest, under the forest cover, but now because of this threat, because that Geodorum densiflorum is acting as an adulterant to Eulopia nuda, this population of Geodorum densiflorum is again declining. So we have collected this Eulopia nuda from the Medghat region, 1 and 2 individuals, and that to be from a very undisturbed habitat. But we have collected many plants of Geodorum densiflorum, but now the population of Geodorum densiflorum is declining because of the similar corm size as well as structure, which is matching to the Eulopia nuda, an anti-cancerous plant. So this is Rhynchostylis retusa. This is the asymbiotic seed germination of Rhynchostylis retusa. You can see how minute seeds are of orchids. So they are growing on the different medias. Then this is Venda tessellata, again an epiphytic orchid. This is the germination of Venda tessellata. Then this is Venda testacea. Then this is Habaneria furcifera. You can see the tuber is growing. Then this is Peristylus densus. So wherever we have found the seed of any species, we used to try it for asymbiotic germination. Then this is Zeuxin stratumatica, a wetland orchid. Then along with the asymbiotic seed germination, we have also tried micropropagation in some of the orchid species. So out of, we have collected 53 orchids in this investigation. Out of 53, we have tried asymbiotic seed germination in 15 to 17 species, and we have successfully developed the protocol for asymbiotic seed germination by using variable medias, variable cytokinin and auxin concentration, along with the variable sugar concentration for the terrestrial orchids. Then we have also tried micropropagation. Here we have used the different parts of that plant for micropropagation. So we have tried the micropropagation in vanilla, cymbidium, Oberonia, and Zeuxin stratomatica. Here you can see these are the photographs of micropropagation. This is the micropropagated plant which is hardened in our polyhouse. Then this is the cymbidium. Here you can see the multiple plants are coming out of the one plant. Then this is Oberonia recurva. Here you can see that multiple plants are emerging out from the base of the main plant. And we have also seen in vitro flowering in this plant. Then this is Zeuxin stratomatica. Here you can see the multiple seedlings are coming out. Along with the asymbiotic seed germination, multiplication of the plant by using lab methods, we have also tried some traditional method, or we have also observed some traditional methods like vegetative propagation. So it was observed that we have also calculated the rate of vegetative propagation in different terrestrial orchids. So here you can see this is the Habaneria foliosa, how luxuriantly that plant is growing, as well as the tubers are multiplying vegetatively, and in a single pot you can see multiple plantlets are coming out. We have also tried reintroduction of the lab plant as well as the plant which we have collected from a threatened area or which is more prone for the destruction, and then we have transferred that plant into much safer area, and you can see the plant is growing very nicely in the next consecutive area. And all those plants which were multiplied in the lab, we have also tried to reintroduce all those plants in the nature wherever it is possible. Then let us move to the conclusion. In this investigation, 53 orchid species were tried for ex situ conservation. In absence of pollinator in our nature, we have also tried artificial pollination. In some of the plant, we found a very good success rate of artificial pollinator. We can say that we became a pollinator for them. And in some of the plant, they were very, very specific about their pollinator. Sometimes we fail to find out what is the reason behind it, that why we are not getting the capsule, even though we are doing the pollination in a nice way. But still we failed in some of the plant. And then plant tissue culture protocol was established for 15 orchid species by applying by using 6 different medias, and then by variable concentration of cytokinins. There was one problem with the families, that some of the plant was interacting very nicely with the media, but some of the plant like variable species of Habanerias, like Habaneria panchganensis, which hardly interacts with the water. So in that case, we have to try multiple things. That sometimes we have used sodium hypochlorite as a bleaching agent so that the plant can interact with the media. Sometimes we have tried suspension culture for the germination. So we found that no doubt the plant takes too much time for the germination on the solid media. Means even though the plants are taking 2 months, 3 months, 6 months for germination, even only for the germination in the solid media, but we found that whenever we have tried suspension culture, within 11 days we found that the seeds are germinating, which were taking 6 months to germinate on the solid media. So we have tried each and every thing for the minute seeds of orchids. The first problem was that the seeds were very minute. How you are going to sterilize them, because the seeds are floating on the water. So how you are going to sterilize them, that was again a problem. So because of this, let us conclude, because of the threat that we have observed while surveying the different areas, so we have tried to put something, or we have tried to conserve some of the plants. It may be a very small work that we can conserve only a limited plants, but we have tried our best to achieve this. So let us talk about what are the problems. The first thing, whatever photograph you have seen till now, 99% photographs are from my thesis. 99% of the flower photographs were taken by me by my guide, Dr. Dongarwar sir. And all photographs are personal photographs, so they are not taken from internet. So let us talk about what were the problem we have faced. So we have visited many localities, and that to be after first rain shower. So whenever excess rain is there, then these plants will come out. So we have visited many forests in the rain shower region. So our vehicle is used to stuck into that area many times. So we were here, you can see that our vehicle is stuck in the forest area. Then this is the condition when we used to visit the Western Ghat. This was the condition, this was the climate. Continuous rain shower is there. In that rain shower, to take a photograph of that plant in the natural condition along with our DSLR. So it was very difficult to manage our cameras along with the collection of the plant, as well as lots and lots of problems were there. Then the another thing is that the plants have adapted itself to climb the tree. So we have to adapt ourselves for collection of the plant to climb the tree. So here you can see my guide along with me is there on the tree to collect the plant. Then here sometime you can also take the help of a tall person. This is my would-be husband, so I have on his back to collect the plant. Sometime you can take the advantage of others' height. So this whole work is completely dedicated to my guide, because my great grandfather, Dr. K.H. Makte, used to say that whenever sir visits the forest for the collection of medicinal plant, and at the time of his research, he always focus on the orchids. So this was my fortunate part, or this I feel very fortunate enough that I have lived that dream of my guide, that I have worked on this family. So here we have, you can see that once we have collected the plant from the nature, the part was not over. Here you can see that you have to have, acclimatize that plant in the poly house. You can see my guide is working with me all the time. At every step, he is sitting along with me for acclimatization or for plantation of these plants. So this is the habitat which is also shared by King Cobra. So this is very threatening habitat, but we have visited such habitats many times. And here you can see that no doubt there is excess rain shower. Many things were there, but there were so many leeches. We have also faced these difficulties. So it was practically impossible to visit alone, being as a girl, into such localities. So this is the team of my guide, who always, or they are always with me at every position, that they have traveled with me even though to the highest peak of Maharashtra, that is Kalsubai area. Twice and thrice we have visited that area. Dr. Valay Thagre sir, Prashant sir, late Shrikant sir, then my husband, my brother, then Mrunal Kale sir, then Kavle sir, Fuljale sir, Soham, Labane sir, and there are so many colleagues of mine. They have visited along with me for the forest area for the collection. So they are always as a back support for me while doing this work. So that's all from my side. If you have any questions, the session is open. And thank you from these plant sites.
Thank you so much, ma'am. Friends, if you have any question, please ask. Any question or any feedback?
Very nice and excellent work, Dr. Uma madam.
Thank you, sir.
Thank you.
I have dared to speak in front of you all.
Yes.
I'm so sorry about the technical difficulties, but
No, it's a technical difficulty. There is no solution. Actually, I want to ask one question.
yes, sir.
When we go for artificial pollination,
Yes, sir.
we collect that anther or pollinia to stick over the stigma.
Yes, sir.
Okay. So what time is required to stick that Polonia over the stigma, or is there
Sir,
any required? Yes, yes.
Sir, mostly it is at the morning time or it is at the evening time, because we have also tried one experiment in one of the species, that what is the specific time for the pollinia and what is the specific time at what time the stigma is more receptive. So it is mostly at the morning and evening time. And we have also tried how many old should be the stigma, how many old should be the pollinia. So we have also tried the variable combinations. So it is mostly the morning time or evening time which is best suitable for the pollination.
Okay, okay. Do we need to transfer it immediately?
Yes, sir.
As
You
soon as-
have to transfer it immediately. We have not stored the pollinia for future use. You have to transfer it immediately.
Okay, okay. Thank you so much, madam. Thank you.
Okay. Thank you, sir.
Dr. Preeti, madam.
Madam, I think there are no questions.
Okay, ma'am.
So on behalf of UGC HRDC Nagpur and from all the participants, I extend my thanks to Dr. Uma Thakur, madam, for your nice presentation and informative session. Thank you, ma'am.
Thank you so much, ma'am.
Thank you.
Thanks,
Thank you.
everyone.
Yeah. Thank you, all of you.
Thank
You can proceed for your lunch break. Our next session will start from 1:30. Thank you.
Thank you, madam.
Thank you, ma'am.